We can see your screen, but we cannot hear you.
Do you hear me?
Yes.
Hello, everyone. I'm very glad to see you all and glad to have a part in this conference. And dear colleagues, let me present a result of our research named Human Mesenchymal Stromal Cells, alginate encapsulation, beneficial effect of storage at ambient temperature under hypoxic condition. Human mesenchymal stem stromal cells, MSCs, are promising cell type for biomedical researches and clinical medicine. MSCs can be derived from a variety of sources, possess multiply differentiation ability, provide a modulatory effect. Efficient methods for MSC storage are required for their practical use. Cryopreservation methods are widely used for long-term storage, but have some disadvantages. Expensive equipment, liquid nitrogen, toxic CPAs. Short-term storage at ambient temperature may both solve main problems associated with low temperature preservation and simplify transportation. The purpose of our work was to study the viability and metabolic activity cell cycle of human MSCs under culture and during storage at ambient temperature in form monolayer, suspension, and encapsulated in alginate microspheres, IMS, and the effect of alginate encapsulation on resistance to hydrogen peroxide. The experiments were performed on human dermal MSCs derived after adult donor-informed concern. For alginate microspheres formation, MSC suspension in 1% sodium alginate was extracted through a needle containers to the high voltage generation into 100 millimole calcium chloride solution for solidification. MSCs in form of monolayer suspension and in IMS were stored in sealed container and 22 degrees in alpha mem, supplemented with 10% of fetal bovine serum. Viability and functional state of stored cells were analyzed at 0, 3, 7, 10, and 14 days. For cells cycle analyze we transduced with the PREMA pulse cell cycle sensor, then cultured in monolayer or in IMS with live cell imaging of cell cycle progression with confocal laser scanning microscope. Effect of encapsulation in alginate to such MSCs properties as metabolic activity and resistance to oxidative stress was studied under culture conditions. Oxidative stress was induced by incubation with a different concentration of hydrogen peroxide. Viability measured by FDI titanium bromide staining was sharply decreased in suspension staining at 3rd days of storage. Viability in monolayer was decreased from 7th day of storage. During storage of MSCs in the form of suspension fluid, formation was revealed at times. At the same time, encapsulation in IMS significantly support viability during storage period compared to monolayer and suspension group. Thus, cell viability in IMS was 80% on the 7th day, 60% on the 10th day, and 40% on the 14th day. This slide illustrates data present on the previous slide. Green colored viable cells are more prominent in case of storage in encapsulated form. Encapsulation in alginate microspheres decrease the basal level of metabolic activity before storage starts and then maintain their parameters during storage at 22 degrees. There was a sharp drop in metabolic activity in suspension and monolayer during the subsequent storage. The ability of MSC stored histoplastic and proliferate after 7th day of storage was evaluated. There were a great difference in adhesion capacity, 12% for cells stored in form of suspension, and 64% showed cells which was extracted by IMS dissolving. The cells from IMS proliferated during subsequent cultivation to form confluent monolayer. Therefore, encapsulation in alginate macrospheres supports MSC viability parameter during the short-term storage at ambient temperature. We have shown a positive effect of encapsulation on the storage of MSCs at ambient temperature. And the next step of our work was to find out which mechanism involved in the development of resistant to storage conditions. We have assumed the encapsulation in alginate hydrogen results in mild hypoxia condition for encapsulated cells. If this happens, it increased level of hypoxia-induced factors with which regulates synthesis of some K protein involved in control of metabolic mode, cell cycle, antioxidant defense. They are salt and catalase. We have suggested the oxygen and nitrates come through enhanced diffusion, and if this happens, the level of hypoxia induced transcription factors that affect protein synthesis increase. It is also known that hypoxia storage causes a perturbation of ion balance, oxidative stress, and pH shift. Therefore, the possible role of encapsulation in the resistance to oxidative stress is great of interest. We have revealed in previous experiments that encapsulation results in decrease of metabolic activity. The aim of the next experiment was to study the effect of the MSC's encapsulation in alginate hydrogen on cell cycle performance and cell resistance to oxidative stress induced by hydrogen peroxide. Cell cycle analyzed by cell cycle sensor showed the MSCs in form of monolayer culture were on different paths of cell cycle. In case of availability of sufficient area for growth, at the same time, cells become completely arrested in G1 phase in 48 hours after encapsulation in alginate hydrogel. Cells in monolayer and IMS were incubated with low concentration of hydrogen peroxide during 24 hours. The metabolic activity were evaluated. Both MSCs in monolayer and in IMS showed resistance to low concentration of hydrogen peroxide till the critical concentration hydrogen peroxide. It was detected that encapsulation in IMS increases the resistance. The critical concentration of hydrogen peroxide was 400 micromole for monolayer and more than 500 micromole for MSCs in IMS. The encapsulation of MSCs in alginate increased resistance to low concentration of hydrogen peroxide. Do you see my next slide? Sorry, I cannot see it.
We can see your slide, so you can just continue. And
thank you, Elena.
It's time actually to move on to conclusions,
Yes.
because we are running out of time, so please continue.
The basal level of reactive oxygen species rose, and the level after short-term incubation with high concentration of hydrogen peroxide were analyzed with Encapsulation ROS key. The basal ROS level was equal 32 and 8 early fluorescent units in monolayer and 2.32 FUE in per cell in IMS, and hydrogen peroxide induced level was equal 154 FUE per cell in monolayer and 11.78 FUE per cell in IMS, respectively. This data complete with decreased metabolic activity and cell cycle arrest in encapsulated cells. Therefore, the basal level of ROS was significantly lower in encapsulated MSC compared to cell culture in monolayer. The benefit of encapsulation in alginate microspheres for the MSC short-term storage and transportation under ambient temperature was shown. The decrease of the overall metabolic activity cell cycle process and lower body level of porous and alginate encapsulated MSCs were revealed compared to monolayer. The resistance to hydrogen peroxide action was higher in cell within alginate microsphere. The decrease of the overall metabolic activity cell cycle process and lower body level of porous and alginate encapsulated MSCs, decrease of the overall metabolic activity, temporary cells. That encapsulation helps to preserve cells at ambient temperature and increased antioxidant defense are those mechanisms which can provide resistance to hypoxic storage at ambient temperature condition. Thank you for your attention.
Thank you so much, Alexandra, for excellent report and congratulations on that. And now we have a few questions. What is the size of the microspheres?
We have different sizes of microspheres. It's like from 200 micromole to 800 micromole for hand technique to take capsules.
So, but still all of they are within this range from 200 to 800, as I understand. Okay, thank you. And another question is from Taisia. How did you return MSCs from alginate microspheres to the culture? When you need to check proliferation capacity or something like this, how you can isolate back cells from the alginate?
Can you repeat, please?
How did you return MSCs from the alginate microspheres to check the proliferation capacity of the cells? How you can dissolve alginate capsules?
Thank you for the question. I need to open my presentation again.
Yeah, we can see it.
Can I answer in Ukrainian some words? Мы растворяли капсулы для того, чтобы проверить после растворения капсул адгезию, то есть к пластику, и с помощью адгезии мы проверяли, насколько клетки сохраняют свою жизнеспособность.
The question was how exactly you dissolve alginate to get the cells out. Because you're explaining that, yes, you need to dissolve alginate to check the adhesion capacity of MSCs, to check the proliferation rate, but I guess that Taisia is just asked about the protocol, how you can get those cells out of alginate. So what protocols, what reagents did you use to dissolve alginate?
I'm sorry, I can't answer this question. But no, I told about it with my research advisor, and maybe the next time I can answer this question.
I guess that's like the usual techniques is to use EDTAs just to dissolve the alginate capsules, and it's not a harmful procedure, so it's like it's routinely used, and usually you can use cells after de-encapsulation, so nothing bad can happen during this process. And there are different techniques to get cells out of alginate gels, alginate capsules, and so on. Also, you have another question, Alexander, from Brian Grout. Alginate at 4 degrees, perhaps you used, so what was the, maybe it's a question about temperature, I'm not sure actually. So you use for the storage 22 degrees and that's all, the one?
Yes, we use only temperature 22 degrees, but because it's the temperature of 4 degrees, all enzymes do not work, mitochondria stop working, and this temperature is at hypothermia, low positive temperature, which can cause cold injury. Although controversy, temperature close to 37 degrees, not suitable for transport in closed container, sealed container, because the cells continue to breathe actively, their metabolic activity is kept high, so they are more prone to hypoxia. And we use 22 degrees for it, and we think it's more suitable for transportation from one research center to the medical center.
Thank you so much for your report. And also I can see on your slide that you actually mentioned that citrate solution was used to dissolution of